Hello, my name is Courtney Oldchief, and I am a student of wildlife and fisheries at the Salish Kootenai College. I am from Browning, Montana, and I am a member of the Blackfeet people, and our cultural name is the Nitsitapi, the real people. My people had a close relationship to Mother Earth and depended on her to provide all of their needs. They were hunters and gatherers, so plants and animals provided what they needed to survive, such as medicine, food, shelter, and clothing. I was taught to always respect the earth growing up because she sustains all life. I came to Salish Kootenai College in 2020 and started with my academic work. My career goals will be to work with the wildlife out in nature and also in research to help the animals and the natural world that they live in. The research project that I started on in the Life and Sciences department has been focused on the Flathead Lake and different types of microbes that live in the soil. The health of the lake and the fish depends on the health of the microbes in the lake. Knowing what balance of bacteria there are in the lake helps us to understand the biogeochemistry of the water. Some of our work also looks at the microbes that are normally found in glaciers. Flathead Lake is fed by the Northern Flathead River, which comes from Glacier National Park. As there is more glacier melt in warmer years, we are finding more of the bacteria from glaciers at the bottom of our lake. This is interesting for us to understand how these extremophiles will and have adapted to an environment without ice and with many other mats of bacteria. We are currently writing up our research work to date, and I will be an author on the publication. Future work that I am interested in will be to look at how to examine microbes that impact the wildlife in our area. This is a new course of study, and we are excited to begin this work. I can't talk too much about what that future work will look like until we are funded, but it will be research that is controlled completely by the tribe and carried out at Salish Kootenai College.
Hello,
we won't be sending any of our samples away as we have our own sequencer now to analyze our DNA data. It is so important for Native people to be able to control the research that is done on the reservation.
Where I received my bachelor's in microbiology.
Having the data
after which I left the state for some time.
With the tribe and the Native researchers here is also important to make sure
then I went to Scotland
that it is not abused or lost or used in an inappropriate way.
After leaving Scotland, I really wanted to return home to Montana. I was fortunate enough to secure a position here at Salish Kootenai College and be able to work with the tribal community. Here at Salish Kootenai College, it's very important that we are able to offer our students novel research opportunities. One of the research projects that we have been working on is the microbial characterization of microbes in Flathead Lake. Flathead Lake is the largest body of fresh water west of the Mississippi. It also is on the Flathead Reservation and directly impacts the people that are here. Let's move on to our cellular and molecular biology lab, where a number of this research has occurred. As we walk down the hallway, we're going into the cellular and molecular biology lab. A number of students have been involved in this project over the past 3 summers and have collected hundreds of samples that we are storing in our -20-degree fridge. These soil samples have been dredged from the bottom of Flathead Lake. Upon thawing these samples, students are taught how to isolate RNA from the soil. We're moving into another portion of our lab here where we have an RNase-free table. At this table, students are going to isolate RNA. Why are we isolating RNA? Because we want to know the metabolically active microbes that are in these samples. That means things that are still alive. Once the students have isolated the RNA, they're going to turn it into a more stable form called cDNA using our PCR machine. Once we have that stable cDNA, we're going to target the 16S ribosomal genes. These are critical to the identification of microbial communities. Upon that PCR amplification, we're going to purify those by gel electrophoresis. Again, undergraduate students are doing this entire process. We mentor them through each step so that they are learning every single technique, from being able to go out in the boat and get a soil sample, which is a lot of fun on the lake in summer, to running their own gels and carrying out their own PCR reactions. We're able to do all of this at Salish Kootenai College and more. Let's walk down to our bioanalyzer and sequencer. While we take a walk, let's talk a little bit about the students that I have mentored in the past here. Now, these students are coming from multiple different departments. Of course, we really want our life science students to have as much novel research opportunities as possible, but we also want to help everyone in the STEM community and tribal community here at Salish Kootenai College. And these include students from the wildlife department, hydrology department, forestry, and nursing. We have had several of each of these types of students come in through the years to do their novel research and work on this project in particular. So as we move in here, we're going to see where we have our bioanalyzer. Bioanalyzing our data is important so that we can understand the quality of the DNA that we have. Once we have a quality band of DNA that we know has been amplified for that 16S ribosomal gene, we're able to take that DNA and sequence it here again at Salish Kootenai College on our iSeq 100. Using a sequencing cartridge, we're able to put our DNA library into the machine, have it sequenced, and receive the data of what microbes and microbial communities exist in our lake, all here. That data is then analyzed by these students, the undergraduate students. The mentorship role is very important to me and to all faculty here at SKC. Thank you so much for your time and for listening.
Thank you so much for taking the time to watch my video.